Welcome students. This is for Pharm D students. I've taken a new chapter, Lipids. This is lecture 1 because it's little widerly we have to discuss in this chapter because they are asking many questions, 5 mark and 10 mark. Even in a question they have asked a 10 mark question and a 5 mark question. So we need at least 4 lectures. So this is the first lecture regarding lipids. Some of the things you might have studied in your smaller classes. I have taken some of the past Kerala University of Health Science questions since 2015 just to have a glance on it. So you see here they have asked define acid value and iodine value and give their significance. 5 mark question. These are repeated questions actually. Write a note on preparation of cocoa butter and olive oil. It's 2022 a 5 mark question. Source of active principles and uses of the following. In that they have mentioned castor oil. It's in half question. Agar from other chapter, carbohydrates. And methods of analysis of lipids they have asked for 5 mark question in 2021. And any 5 parameters in evaluation of castor oil. As per IP, Indian Pharmacopoeia, they have asked for 5 mark question. Define iodine value and its significance. Explain a method for its determination for 5 mark question in 2022. They have asked what are lipids. Explain different methods of estimation of lipids for 5 mark question in 2023 March. Describe the preparation of beeswax and shark liver oil, 5 mark question in 2023. Write the production, description, chemistry and uses of shark liver oil, 5 mark question in March 2023. Explain in detail about methods of analysis of lipids, 5 mark question in 2018. Source, constituents and uses of beeswax and wool fat for 5 mark question they have asked in 2017. And you can see they have asked about the principle involved in the determination of saponification value and iodine value of a fixed oil. 5 mark question in 2015. Explain the extraction methods of fixed oil in 2021. Define lipids, examples or explain the methods of extraction of lipids in 2018. Give the source, chemical constituents and uses of chalmogra oil and olive oil. 5 mark question they have asked in December 2023. Cocoa butter separately, not guar gum is from a different chapter, but cocoa butter they have asked for in 2018. Write the sources and uses of wool fat and castor oil and other drugs also comes under in other chapter, but I have taken which one is more specifically in our chapter they have asked. You can see 10 mark question. How much 10 mark question they have asked repeatedly. Write the source, methods of extraction, chemistry and method of analysis of lipid containing drugs for 10 mark in 2019. Give the source, method of production, constituent test and uses of castor oil and chalmogra oil, 10 mark in October 2022. Define lipids, Explain the pharmacokinetical nature of lipids used as in cathartic properties. It's also castor oil, 10 mark question, August 2021. Explain the various methods of preparation, sources, characteristic constituents, uses, and chemical test for castor oil, 10 mark question, June 2016. Define lipids and classify them. Give the different methods of extraction and analysis, 10 mark question, in July 2017. So you see that they have, even I have seen specifically some year of question, they have asked one 5 mark and one 10 mark in this chapter. So we will have, I can give some idea about this chapter in 4 lectures, but still the student have to elaborate, detail, reference and preparation of their notes is very important. First we will see the definition in a small manner, not to have, there are many definitions, many explanatory definitions are there, but still it is possible you can refer and do the details. But I have given for examination point of view, lipids are broadly defined as any fat soluble or lipophilic, a simple definition, naturally occurring molecules such as fats, oils, waxes, cholesterol, sterols, fat soluble vitamins like A, D, E and K, monosaccharides, disaccharides, trisaccharides and phospholipids. There are many other categories also there in definitions, but this is a simple definition which a student can write regarding starting is about lipids. What are lipids? And classify them. An analysis, if they ask a question in 10 mark, you can start with the definition with giving this. And you should write the main biological function of lipids. Biological. In a living organism. Biological function. Its number 1 is energy source. Number 2 is acting as structural component of cell membranes. And number 3, participate as an important signaling molecules. Even every DNA, RNA and also signaling or genetic signaling characters, signaling molecules, they are very important part you should mention. This is the function of, biological function of a lipids. And when we see classification of lipids, when we see the classification of the lipids, on the basis of the nature of the product obtained on hydrolysis, first we can classify them. Simple lipids, compound lipids and derived lipids. When we talk about simple lipids, upon hydrolysis yield one or more fatty acid and an alcohol. You know that fatty acids, it is an actually, when you say lipids are esters of fatty acids. So when you hydrolyze, simply if you get an acid and an alcohol, then we can say it is a simple lipids. Compound lipids upon hydrolysis yield, in addition to fatty acids and alcohol, compounds such as phosphoric acid, sugars. So that is compound lipids. Derived lipids, you can see, do not contain an ester linkage, but may be considered to have been derived from natural occurring esterified materials. Esterified materials. Example, steroids, vitamins, plant pigments. So clearly a student can write this very simple definition and the classification of lipids. Fixed oils and fats, when we talk about, both fixed oils and fats are termed as triglycerides or triacyl glycerols. These are the reserve food materials of both plant and animals. The difference between fixed oils and fats is their physical state. This the student should know. Those which are solid between 15.5 to 16.5 degrees centigrade are termed as fat, while those which are liquid between 15.5 to 16.5 degrees centigrade are termed as fixed oil. Fixed oils and fats are esters of fatty acid, just now I told, with glycerol, that is triesters of glycerol. Glycerol is a trihydric alcohol containing 3OH group, which combines with three same or different fatty acid, can combine with same type of acid group or a different type of acid group also possible according to the long chain carboxylic acid. So to form triglycerides, and three molecules of water will be removed when a three molecule is attached. You can see this structure. Glycerol 3OH, when you are combining with same molecules of acid with R is just mentioned, we can change it, but this is just a general formula. So when you add an acid, it's immediately removal of water molecule. You will get ROH plus ROOH gives RCOOR. This is the structure. This is ester, glycerol, alcohol, fatty acid, acid, then combine forms an ester RCOOR, triglycerides, by removal of one water molecule. You can see very clearly the formula. When three molecules of same fatty acids combine with glycerol, then the product is called simple glyceride. When two or three different fatty acids combine with glycerol, then it is known as mixed glyceride. You can see there is two examples. Here glyceride tristearate, or you can say tristearate, simple glyceride. Why? Because all the glyceric acid which you combine with the glycerol is same. That is 16 to 17 carbon atom containing long chain is fixed. And here also the same thing, 17 carbon atoms. Similarly, all the three OH is removed and get attached. And you can see here in glyceryl palmitosterolate is that, or stereolate, in that difference is there. The first molecule is having 15 carbon atoms, second molecule is having 13 carbon atom, long chain without any double bond. But the third one, you can see it is an unsaturation. Double bond is also there, and you have more than 16 carbon atoms are there. So that's why the difference of acid when it is attached in the ester linkage, then they can called as mixed glycerides. So these two examples, students can remember what is a simple glyceride and also a mixed glyceride. Fatty acids are carboxylic acid with a hydrocarbon side chain. These fatty acids bear even number of carbon atoms. Generally, natural fatty acids are naturally containing fatty acids with 14 to 24 carbon atoms. Fatty acids may be saturated or unsaturated. Saturated means actually these do not contain any double bond as shown in the picture. They are all single bond, pi bond system. And when we come to unsaturation, you can see the double bond system will be there. And saturated means, sigma bond. Unsaturated means pi bond. I just mistakenly told that is saturated fatty acids is sigma bond, strong bonds. There will not be any pi bond of double bond. And you can see unsaturated fatty acids, these contain one or more double bond as shown in the picture. When a single double bond is present, then they are called as mono unsaturated fatty acid. If they are having many, you can say poly unsaturated fatty acids or PUFA is a multi double bond are present. Then you can say it is poly unsaturated fatty acids. So when you come to an example of saturated fatty acids, we should have a writing of some examples you should know. For example, butyric acid, which contain, you can see, more than 4 carbon atoms with a long chain of COOH. You can see the formula, and it is origin is from butter fat. Caproic acid, you can see it is having 6 carbon atom of long chain acid, and the melting point is minus 2 degrees centigrade. Palm kernel oil is the origin. Caprylic acid, it's of 8 carbon atoms, long chain. Formula is very important. And also you can see the melting point is 17 degrees centigrade, and it is present or origin in coconut oil. Caproic acid, you can see 10 carbon atom long chain, and 31 degrees centigrade is the melting point, and it is present in palm oil. Lauric acid, it is 12 carbon atom long chain acid, which is having a melting point of 44 degrees centigrade, which is present in coconut oil, or origin is in coconut oil. Myristic acid, it has 14 carbon atom long chain, and it has a melting point of 54 degrees centigrade. Present or origin is palm oil. Palmitic acid is have 16 carbon atom long chain acid of melting point 63 degrees centigrade. It's the origin of rachish oil, sesame oil. Stearic acid, which is having 18 carbon atom long chain acid of 70 degrees centigrade, is a melting point, which is an origin of rachish oil and sesame oil. So these examples, very simple for the students to remember about these oils, because which is very commonly which we are using for various food preparations. When we come to unsaturated fatty acids, these are also very important examples you should write. Palmitoleic acid, which you can see an unsaturation, one double bond is there between 7 to 8 carbon atom, 16 carbon atom long chain fatty acid, which is an origin of cottonseed oil. Oleic acid is an unsaturated fatty acid, which has also one double bond in between 9 and 10 carbon atom, 18 carbon atom long chain fatty acid, which has 16 degree centigrade as a melting point, and the origin is corn oil, sunflower oil. And you can see that linoleic acid, which has 2, one is 6 to 7 and also 9 to 10, like a conjugated double bond system. It has 18 long chain fatty acid, linoleic acid, which is having minus 5 degrees centigrade as a melting point, which is an origin of sesame oil and sunflower oil. Linoleic acid, which is having 3 conjugated system of double bond, 3 double bonds, unsaturation, 18 long chain carbon atom fatty acid. It is one which is an origin of linseed oil and soybean oil. And next we can see that arachidonic acid, which is having 4 conjugated system of double bond system, 4 double bond unsaturation, 20 lengthy long chain carbon atom fatty acid, which is origin most of the it's not shown very particularly because it is present in many of the some of the oils it is present. So that's why arachidonic acid is also an example which is added as an unsaturated fatty acid. Now classification. Oil, already we have classified according to the hydrolysis. Here fixed oils and fats can be classified based on the sources also, from animal source, plant source, which source also can. Plant sources, you can see it can be classified as a fat from plants. These are solids at temperature between 15.5 degrees centigrade to 16.5 degrees centigrade. Example, cocoa butter, nutmeg butter, palm oil, etc. Drying oils. These oils, upon exposure to oxygen, harden to form a tough solid film due to cross-linking of their components. Example, linseed oil, poppy seed oil, walnut oil. You can have an example of these natural sources. Semi-drying oils, these partially form a tough solid film upon exposure to atmospheric oxygen. Example, castor oil, mustard oil, sesame oil, and etc. And now non-drying oils are also there in the D. These do not harden at all upon exposure of oxygen. Example, olive oil, peanut oil, almond oil. So students should know about this difference. They should mention when they ask classification, not only by hydrolysis, you should write at least the source. By means of source, it's interesting to classify them by giving some examples which we know as a natural sources. And animal sources. Now plant sources we have shown. Animal sources, fats, example lard and sweat. When we see oils, next one is oils. Oils, example cod liver oil, shark liver oil. We can give the two examples which are mostly used. Lard means it's made up of 100% animal fat, usually from pork, that has been separated from the meat of the pork. This is the structure, looks like white. Sweat is a raw, hard fat of beef or lamb or mutton. You can see this is the color. You can see that is slightly yellowish outer layer, and it is the fat we can take from the meat directly. So you can see the properties contain certain property common for the both fixed oils and fats are as follows. Just we have to have a discussion. What is that? They are greasy to touch, both of them. Insoluble in water, but soluble in organic solvents like chloroform, petroleum ether, and benzene. Being lighter than water, they float on the surface. They are non-volatile in nature and cannot be distilled. They undergo saponification reaction. What is saponification? That when boiled with alkali, the product, that means we are talking about the fats and waxes or fats and oils, the product forms an alkali salts when it is boiled with alkali of fatty acids, termed as soap, along with a glycerol molecule. Basically, saponification represents the alkaline hydrolysis of fats and oils. You can see the structure or a reaction, how a sodium hydroxide soap formation. A simple triglyceride we have taken for the reaction, and when you see it forms glycerol, and the soap, the salts, sodium salts of the triglycerides is formed, that is the soap. Glycerol is separated. That's what the reaction which we explain. And the sixth point is fixed oils and fat turned rancid also. They not only form soap with alkali, they have another reaction. They may turn rancid due to microorganism deterioration, gives the unpleasant odor. Triglyceride made up of unsaturated acids are more susceptible to rancidification. So unsaturation is there, means it's possible for rancidification very fast, getting deteriorated. So saturated oils are good for storing and to use for many purposes. Avoid this rancidity. We can avoid even adding preservatives. Now we will come to the extraction process. They have asked many times the question, extraction, specifically for each individual drugs also it is needed, crude drugs. But this is common needed. Suppose if they are asking question, extraction. Fixed oils and fats are extracted from both plants and animal sources by the following three techniques. One is rendering. Rendering means animal tissues from which fat is to be extracted is minced and heated with or without water. The fat melts, which is then suitably collected. An example of shark liver oil, cod liver oil, which is taken from the fish, we can say raw material of the fish is cooked and pressed and decanted, and centrifuge and solid fraction can be separated, and crude oil again refined, and you can get the fish oil. We are not going in much detail, just an outline which we can express for this extraction of rendering method. Pressing. In this technique, seeds containing oils are initially crushed between the steel rollers and then fed into hydraulic press. Like there are many hydraulic presses and also rolling steel roller mills are also there, like how we are taking coconut oil, groundnut oils locally which we are making. This I have taken one example of olive oil. Washed olives seeds have been grinded, and then we can say malaxer. The malaxer means with the not matrix, you can say the residue or the mark with a solid with an oil of the crushed one of olive is pressed for extraction, and olive pomace is obtained, and then this oil and vegetable water can be removed by decantation. Olive can be separated from the water. The water can be removed. This is just a picture, but it's not an instrument which is shown very clearly. There are instruments which can clearly, they can separate the water and the fixed oil separately. Solvent extraction, chemical extraction, commonly which we have studied. This is for fixed oil also can be applied. But in this residue, after rendering and pressing, the two process is treated. The suitable solvent can be used are petroleum ether and benzene to extract the remaining fat or oil. So you can see this picture, just an outline, how water can be, solvent can be sprayed as a vapor over a solvent distribution, or even you can use, apart from the water vapor, even you can use other solvents also. This is just for a solvent which how you do an extraction with water. Other than that, suppose after you can use even solvents can also be used. This picture is only for water it is shown, but just this instrument I have taken, this instrument can be used for solvents like petroleum ether, benzenes. Benzene is little toxic, but you can go for other solvents. I have given examples also. This procedure makes us to or use the solvent that can successfully extract oil from seeds or other raw material by dissolving them. The solvent that can be used is include even hexane, acetone, ethanol, chloroform, and propane. However, the most common used solvent is hexane. The colorless flammable liquid is considered as good solvent for oil. This is a study reported very clearly that N-hexane can recover 37.03% of oil from a sesame seeds, while chloroform can only recover 6.73% and acetone can only recover 4.37%. So N-hexane is the best one to recover after any sesame oil. They are seen in the sesame seeds. After rendering and pressing, and making the treatment with the N-hexane extraction or a chemical extraction. It was successful for recovering the oil which may be present in the mark. Identification test: fixed oils and fats can be detected by presence of glycerol, which is produced upon their hydrolysis. See, very simple chemical test. You needed to write and remember as a, you have to write clearly. Not writing any chemical, they will not give you mark because it's a simple 5 drops of fixed oil or a fat, 1 ml of 1% copper sulfate solution, 5 drops of 10% sodium hydroxide solution. You will get a clear blue solution, so indicating the presence of glycerol. Next test is 5 drops of fixed oil or fat with 0.5 grams of sodium hydrogen sulfate. You will get a pungent odor of acrolein, so indicates the presence of glycerol. So these are all the things which we have discussed for the first lecture. Next lecture we will discuss about the analytical analysis techniques which you are doing something in the lab also. So that will be helpful for the next lecture and we will go for the continuation. Thank you. Thank you for listening. So you just listen to the class and prepare your own notes. This is a very simple starting for lipids.